Every cell is involved in making large molecules that are needed not only to build the body parts of organisms, but also to maintain the biochemical processes that keep them living, such as communication, transforming energy, and relaying genetic information. These large molecules are called biomacromolecules. So bio means living, macro means large. So these are the large molecules that living things consist of. Biomacromolecules are grouped into four main classes based on their chemical composition and structure, and these are carbohydrates, lipids, proteins, and nucleic acids. Autotrophs can build all these organic compounds from the materials that they take in from their surroundings. So for example, seaweed, eucalyptus grass, and microscopic algae all produce simple sugars through photosynthesis. They can then use these simple sugars to build other kinds of compounds such as sucrose, starch, and cellulose, which help to form the structure of these organisms. Heterotrophs, on the other hand, have to make their own biomacromolecules from existing organic compounds. They take in a range of organic compounds such as protein in their food. They then break these macromolecules down into simpler substances, such as amino acids, in the process of digestion. These are then synthesized or built back into the kinds of organic compounds that are required by the organism. All macromolecules are chains of small molecular units called monomers, and when monomers are linked together, they formed polymers. So for example, consider this shape here to be a single unit of one of our biomacromolecules, and that is referred to as the monomer. Mono being singular or one. Now if you were to get another monomer and join it to that first monomer and keep doing this in a chain, you end up with a polymer, where poly means many. And this is true for all of the biomacromolecules: carbohydrates, lipids, proteins, and nucleic acids. So if you learn about the base unit, or the monomer, for each of these biomacromolecules, it makes it a lot easier to understand the polymer that they form. Cells use a chemical reaction called dehydration, also called a condensation reaction, to link monomers together. It's called a dehydration reaction because water is removed when a bond is formed between two monomers. And by repeating this reaction, cells can build extremely large polymers consisting of thousands of monomers. So here is a chain of monomers that have all been linked together in dehydration reactions, and here is another monomer that's just about to link onto the end of that polymer in a dehydration reaction. So in a dehydration reaction, an enzyme removes an OH, or a hydroxyl group, from one molecule and a hydrogen atom from another molecule. And what ends up happening is that forms a water molecule, and a bond is built between your two monomers. So they would look like this when they're linked. The reverse reaction is called hydrolysis. This is where enzymes break the links between monomers, and water is used in this process. So again, here is our polymer chain here, which can then be broken down into smaller units. So down below here, you have two monomers which are joined together through the dehydration reaction we just looked at. And in a hydrolysis reaction, you have to replace the water in order to form monomers with a hydroxyl group and a hydrogen on either side of your monomer. Just like that.